Perception. Understanding how sensory information is detected, segregated, and transmitted is only the first step in understanding how animals are able to interact with their sensory environment. Sensory signals that travel through the central nervous system must be recombined in a way that is meaningful to the animal. This connection between sensory input and the analysis of this input describes the relationship between sensations and perception. Sensations are produced when physical stimuli, e.g. light or sound waves, activate sensory receptors, which then send neural signals through specialized circuits. Perception is the interpretation of these signals, which occurs when sensory information is processed, organized, and filtered within the central nervous system. The distinction between sensation and perception is illustrated in Figure 2.12. Sensory receptors detect a series of black dots and random shapes on a white background, with little variation across the two-dimensional image. In contrast to these sensations, most people perceive a spotted Dalmatian with shadows in the background. This occurs because the visual system groups smaller elements into larger objects, which are then recognizable to most humans. Figure 2.12. A series of black and white dots may be perceived as an image with identifiable shapes. So you can see the dog. Psychophysics. The study of the relationship between sensations and perception is called psychophysics. Psychophysics attempts to explain how the physical characteristics of a stimulus are interpreted by an organism. Psychophysical experiments in humans are conducted by asking a participant to indicate when they see a light, hear a sound, smell an odor, etc. The setup is familiar to anyone who has undergone a hearing test. In this clinical exam, tones of different volumes are played through earphones to each ear, and the patient indicates when the sound is audible. The same basic setup is used in experimental labs that test responses to stimuli and other modalities. Naturally, this is more difficult to study in animals, which cannot make verbal responses. But experimenters have figured out clever ways to get yes-no answers from animals in the lab. The simplest way to do this is to train animals to make one response when a stimulus is present and another when it is absent. By varying the intensity of the stimulus, the researcher can determine the point at which an animal no longer perceives the stimulus. See Figure 2.13a. Blau, 1956, set up one of the first psychophysical experiments using this procedure to test how well pigeons could detect light after spending different periods of time in the dark. As shown in Figure 2.13b, the threshold for detecting light dropped as the dark period increased. In other words, the birds were more sensitive to light the longer they stayed in the dark. This lowered threshold for detecting light that occurs under reduced illumination is called dark adaptation. Dark adaptation takes a few minutes to develop because it involves a series of neurochemical reactions in the eye. The mechanism evolved in humans and some animals, providing these species with the ability to see during the day and at night. Dark adaptation provides clear evidence that perceptions are not a direct reflection of sensations. Individuals see different aspects of their environment under altered lighting conditions. Indeed, the same physical stimulus may be perceived in one context but not another. Like vision, perception in other sensory systems is also context-dependent. For example, subtle flavors are easier to detect when an individual is hungry than when they are sated. Figure 2.13, Experimental Design A and Data B from a study examining light perception in pigeons, Blau, 1956. Pigeons are placed in an operant chamber with a pecking key on either side of a food magazine. Over a series of trials, they learn to peck the A, yes key when a light is turned on above the food magazine and to peck the B, no key when no light is turned off. As the light intensity decreases, birds respond less on the yes key, indicating that they can no longer detect the stimulus. This threshold to detect the light declines, i.e., birds are better able to see the light when they are placed in a dark environment prior to testing. Okay, so in A, you can see the bird, and you can see the stimulus as whether the light is on or off. and there's a food magazine, and then the bird will tap A or B with their beak, depending on whether the light is on or off, to get food. And then you can see the data in B. On the x-axis is time in the dark in minutes from 0 to 60 minutes, and on the y-axis is threshold to detect light stimulus, luminance, from 0 to 7. Okay, so as the time in the dark increases from 0 to 60 minutes, it takes less light for the bird to see that there is light shining and then tap the correct key for that. So from 0 to 20 minutes, the luminance must be 6 for them to detect that the light is on. And if you go all the way to 40 to 60 minutes, the luminance only needs to be around 4.5 for the bird to detect that the light is turned on, all the way down from 6, as they spend more time in the dark before the light is turned on. Deciphering the soprano and bass lines may be simple in an unaccompanied choral piece, but near impossible in a large-scale opera with full orchestra, and a light brushing of the arm is likely to be interpreted, perceived, in very different ways if it comes from a close friend rather than a complete stranger. The natural environment provides infinite examples that perception is altered under different stimulus conditions. Think about camouflage, in which structural adaptations allow a species to blend in with its environment. The effectiveness of camouflage can be measured by testing whether the same animal is more difficult to detect in different environments. Kettlewell, 1955, used this approach with the peppered moth, Biston betularia, which is either melanic, black, or lightly speckled, peppered. He placed each type of moth on dark or pale tree trunks and recorded how many became prey to local birds. As expected, melanic moths on light backgrounds and light moths on dark backgrounds were more conspicuous, being gobbled up quickly by the birds. Kettlewell's report helped to explain why the once abundant speckled moths were replaced almost completely by the rare melanic form in certain areas of Great Britain from the mid-19th to the mid-20th century, so the mid-1800s to the mid-1900s. The treed areas in which the melanic form became dominant were darkened by industrial soot. Under these conditions, the light-peppered moths became easy targets for predators, reducing the survival and reproduction success of this subgroup. Fortunately, Britain eventually reduced its use of coal and enforced stricter environmental laws, so the speckled moth morphs increased once again. Psychophysical experiments and the natural environment equivalent, camouflage, reveal that the ability to detect a sensory stimulus depends on the context. The same principle applies to detecting differences between stimuli. A just noticeable difference, JND, the amount by which two stimuli must differ so that the difference can be detected, is not an absolute value, but a relative one. As the intensity of a stimulus increases, so does the intensity that produces a just noticeable difference. Thus, if a dim light in a dark room is turned up a small amount, the change is immediately noticeable. In contrast, a very bright light needs a much larger illumination boost to produce a perceptual change. The ratio between current stimulus intensity and the intensity change required for a just noticeable difference is called the Weber fraction in honor of Ernest Weber, 1795 to 1878, the physiologist who characterized this relationship. Just noticeable difference makes sense if you consider that the function of perception is to interpret sensory input. What an organism needs to know is how particular stimuli stand out or differ from other stimuli. The best way to do this is to design a system that calculates the relationship between stimuli. In this way, the system can process sensory input that may vary over many orders of magnitude without losing sensitivity in the lowest ranges. Most importantly, just noticeable difference provides a striking example that the perceptual system does not reflect the absolutes of the sensory world. Sensations transformed to perception. Given that sensations do not map directly onto perceptions, the central nervous system must transform sensory input into messages that are interpretable to the animal. In other words, sensory information that is segregated by modality e.g. auditory or visual, and then coded for stimulus dimensions, e.g. intensity or duration, must be recombined into a perceptual whole. This translation of sensations into perceptions is accomplished in successive stages along the sensory pathways. Recall that sensory information is transmitted from receptors to cortical regions through specialized circuits. Each pathway has relay nuclei, where signals are transmitted from one cluster of neurons to another. Sensory information is integrated at these relay points and is also combined with signals arriving from other brain regions. This general organization of sensory processing is shown in Figure 2.14. Figure 2.14, route of auditory impulses from receptors in the ear to the auditory cortex of humans, left panel. The cochlear nucleus receives input from the ipsilateral ear, the one on the same side of the head. Information is then sent bilaterally to the superior olive, inferior olive, and medial geniculate nuclei. The medial geniculate nucleus in the thalamus sends auditory signals to the primary auditory cortex, right panel, located in the superior temporal cortex. Okay, so in the left diagram, you have the auditory cortex, the inferior colliculus, the signal from the ear, and the cochlear nucleus. You also have the medial geniculate, the superior olive, and then the signal from the right ear. Okay, so they're all kind of headed from the ear to like the center of the brain. Which you can see from the side view in the second diagram is the primary auditory cortex. Okay, so it's like on top of the brainstem near the back of the brain. That's where it's being sent to from the ear. To understand how information is processed at each stage of the circuit, scientists implant electrodes in groups of neurons at each relay station and record how these neurons respond to different stimuli. The visual system again provides an excellent model for understanding this process because neuronal responses to visual stimuli have been characterized in a variety of animals. Most of the sensory receptors in the eye project to a region of the thalamus called the lateral geniculate nucleus, LGN. Sensory receptors in the retina and LGN neurons both fire at rapid rates when small dots of light are presented to the eye. But this does not mean that they are performing the same function in terms of visual processing, or that visual signals are simply transferred from the eye to the cortex through the LGN. Rather, the LGN organizes incoming visual signals that arrive from the retina before transmitting this information to the cortex. First, input from the right and left eyes are separated into different layers of LGN neurons. More specifically, each LGN receives input from both eyes, but fibers from the ipsilateral, same side, and contralateral, opposite side, eyes project to defined regions of the LGN. This organization is even more detailed within layers of LGN neurons, in that each point on the retina sends axons to a corresponding location on the LGN. Thus, sensory neurons that are adjacent to each other in the retina are also adjacent to each other in the LGN. This produces a spatial mapping from the retina to the LGN, a truly amazing feat of organization, given that each LGN receives input from approximately 1 million retinal fibers. Finally, the LGN regulates information flow from the retina by integrating these messages with signals from other brain regions, most notably the cortex. Indeed, there is a much larger flow of information from the cortex to the LGN than the other way around. This suggests that the LGN must be performing some kind of processing that modifies and refines sensory signals about the external world. A separate visual pathway that bypasses the LGN has a different role in visual perception. See Box 2.2. Box 2.2, blind sight. The primary visual cortex is the first cortical area to receive input from the retina via the LGN, so it is not surprising that damage in this region produces blindness. The damage may be caused by stroke, brain injury, or surgery to remove a tumor. Patients with these conditions report no visual perception in the visual field that projects to the damaged area. Experimental lesions in animals appear to confirm this phenomenon of cortical blindness, at least until the late 1960s. Around that time, researchers began to examine the function of visual pathways from the retina to the cortex that did not travel through the LGN. In a clever experiment with golden hamsters, Mesochrysatus auratus, Schneider, 1969, showed that lesions of the visual cortex rendered animals unable to discriminate between visual stimuli, lines of different orientation, but did not impair the ability to turn toward a food reward. Lesions of the retinal fibers that project to the superior colliculus produced the opposite pattern. These animals could not locate a food reward, but were able to solve a visual discrimination task. The explanation for this dissociation of lesion effects was that hamsters, and probably many other animals, had two visual systems, one for identifying the where and one for identifying the what of visual stimuli. Many people argued that the orienting response in humans had been lost or become dormant over evolution as cortical systems became dominant. A small portion, approximately 10%, of retinal fibers project to the superior colliculus in humans, but the function of this where system appeared to be vestigial. The hypothesis was disproven in a dramatic demonstration of residual visual function in a patient with cortical blindness, 1976. DB had a large portion of his right visual cortex excised to remove a malformation. As expected, he reported no vision in his left visual field after the surgery. So it seemed outrageous to him that an experimenter would ask him to locate an object in his left visual field by moving his eyes to the object's position, but trial after trial showed that DB performed significantly better than chance on this task. Weisskranz coined the term blind sight to describe this paradoxical ability of cortically blind individuals to locate a visual stimulus. Subsequent clinical reports confirmed this phenomenon in other patients, although the majority of individuals with cortical blindness do not demonstrate blind sight. Nonetheless, even isolated reports of blind sight help to confirm a functional role for the superior colliculus in human vision. They also provided some of the first evidence for perception in the absence of knowledge of awareness. Blindsight patients repeatedly claim that they cannot see a stimulus. When pressed to make a choice, however, they can point to the stimulus location. These individuals are surprised, even incredulous, when their correct choices are revealed to them. Knowledge of perception of a stimulus, therefore, can be dissociated from the perception of that stimulus. LGN signals that arrive in the primary visual cortex target one of three types of cells. Simple cells respond best to lines of a particular orientation, complex cells to a particular direction of movement, and end-stopped cells to corners, angles, or bars of a particular length moving in a particular direction. In addition, the visual cortex organizes information into separate paths for color, shape, and motion. As sensory information is transmitted to higher brain regions, therefore, neurons at each stage respond to increasingly complex stimuli. The processing of sensory information does not stop in the primary sensory areas. Sensory signals continue to be organized, regulated, and integrated as information is transmitted to other parts of the cortex. In mammals, one of the most interesting, at least in terms of sensory perception, is the orbitofrontal cortex, OFC. The OFC receives direct and indirect connections from all primary sensory areas, suggesting that it has an important role in sensory integration. One proposal is that the OFC assigns motivational significance to sensory stimuli, allowing animals to make decisions about how to respond to these stimuli. In sum, sensory processing occurs in stages, with neurons responding to more and more complex aspects of the environment as information is transmitted to higher brain regions. This general plan of sensory processing is consistent across sensory modalities and across a variety of animals, providing strong evidence that the process is preserved over evolution.